Hey Mustangs, in this video we're going to do a quick recap of cellular respiration stages 2 through 4. So first, before we get started with those stages, we have to remember the parts of the mitochondria to help us really understand what's going on here with stages 2 through 4. So with the mitochondria, with each mitochondria, remember a single one is a mitochondrion. So when you're looking at it, it actually has two membranes. So you have your membrane-bound organelles, and this one's special along also what we're going to see with the chloroplast because it has two membranes. So you have an outer membrane, and then inside that you have an inner membrane. So two membranes that make up the mitochondria. The outer one is your normal, what you think of when you think of a membrane. On the inner one, it's kind of wrinkly and folded. So the inner membrane has a whole bunch of folds as part of it. We call it the cristae. And those increase surface area, which allow more of the reactions that we're going to see happen during stages 2 through 4 occur. So let's go ahead and take a look. The second stage is where we're starting here. So first stage, remember, was glycolysis. That already happened. So all the stuff that happened with glycolysis, remember, that leads right into our second stage here. So remember, we're looking at a metabolic pathway here. So one step leads into another. So we had our first stage, which was glycolysis, and that pathway leads into our second step here, which is going to be pyruvate oxidation. So here, when you're looking at our mitochondria, where is that going to occur? Inside the mitochondrial matrix. So the inner part, so once you go past the inner membrane, the inner part is the mitochondrial matrix. The space in between the two membranes is the intermembrane space. Okay? That's also going to be important as well. So let's go ahead and take a look at pyruvate oxidation. So in glycolysis, we took 6-carbon glucose and went over a series of 10 reactions, converted that into 3-carbon pyruvate. 2 3-carbon pyruvates, all right? So we made 2 pyruvates by the end of glycolysis. Now we're going to take each of those pyruvates and we're going to turn those into acetyl-CoA. So acetyl-CoA, you're actually tearing off one of the carbons and you're adding another group in its place. So when we look at the overall reaction here, we're going to find that the outcomes will be, we will create 1 carbon dioxide molecule, 1 NADH, 1 acetyl-CoA, and that acetyl-CoA will go on to stage 3. Now, these numbers that we're looking at here are for each pyruvate. So one pyruvate will make this. The second pyruvate will also go through it and make these same things. So in the end, if we looked at both of them together, we made 2 carbon dioxides, 2 NADH, and 2 acetyl-CoA. So how does that look? Well, here is our pyruvate. Right there, highlighted in gray, there's a carbon and 2 oxygens. Well, that should click something right there. A carbon and 2 oxygen is your carbon dioxide. So carbon dioxide is going to be removed, and when we remove that, it gets replaced with this right here, coenzyme A. So now we have acetyl-CoA. So we have an acetyl group and then the coenzyme A attached here. And all it did is, you can see, it replaced this carbon and oxygens that we find at the top right here. All right, next step is going to be the citric acid cycle, also known as the Krebs cycle. So a lot of these have double names here. So this one is citric acid cycle, or Krebs cycle, means the exact same thing. So if we look at the citric acid cycle, this is going to actually be 8 steps. So over a series of 8 steps, what we're going to do here is we're going to take that acetyl-CoA and we're going to get the remaining energy out of it. So now if we go back, we have to kind of go back to glycolysis here. We had the glucose, which had the energy. At the end of glycolysis, it got turned to 2 pyruvate, so we still had some, we have energy in there. That pyruvate in the previous step, stage 2, we saw got converted to acetyl-CoA, so there's still energy in there. We got to get the remaining energy out. So the Krebs cycle, or the citric acid cycle, is all about getting that energy out. So what we're going to see happen here is a lot of that energy is going to be transferred into electron carriers. So those electron carriers are going to be huge in the next stage. So here we're on stage 3. Stage 4, those electron carriers are super, super important. So we see acetyl-CoA goes in, we break off that coenzyme A, and a series of 8 steps are going to happen here. And in the process of that, the outcomes for each acetyl-CoA, remember there's 2 of them, you're going to make 2 carbon dioxide molecules, 3 NADH, 1 ATP, and 1 FADH2. So that's going to be the outcome of the Krebs cycle. If you look at the first step is actually taking off that the coenzyme A and taking the acetyl group and putting it onto oxaloacetate. And it's really simple. If you just look at the diagrams here, this part, the acetyl group, will be added to the top right here. When you add those two things together, you're going to get citrate. Now, citrate should sound familiar because we talked about how it can act as an inhibitor for phosphofructokinase, which is an enzyme that is used in glycolysis. So this is a very important step right here. So this will help us control our rate of the Krebs cycle or the citric acid cycle. So very, very important right there. Now there's going to be a series, of course, of 8 reactions that go all the way through, and what we'll see at the end, we wind up going back to oxaloacetate at the end of the cycle. So this is our first step here, and then here the next 7 steps, a series of redox reactions that are going to make your NADH. So it's going to take the energy out of the acetyl-CoA and put it into your NADH and your FADH2, and of course make 1 ATP in the process, which isn't much. So we still haven't made a lot of ATP at this point. Alright, so here is the whole complex set of the 8 reactions that we see going on. So here we said it was our first step, adding the acetyl group onto the oxaloacetate. Go through a series of steps. Here we create 1 NADH, here's another, here's our ATP, here's our FADH2, and then finally our NADH. Okay? And then of course our carbon dioxides that are also made in the process. Alright, so at this point, how many ATPs have we made? And that includes stage 1 glycolysis, stage 2 pyruvate oxidation, and stage 3 the Krebs cycle or citric acid cycle. Okay? So altogether, only 4 ATP. That's all we've gotten out at this point. Now you might be thinking, wait, we made 4 ATP in glycolysis. Well, remember, we only net 2 because while you made 4 in glycolysis, you actually used 2 as well. So we say we net 2. And then here we saw in the citric acid cycle or the Krebs cycle, we made 1 ATP per acetyl-CoA. So that's all we've gotten at this point, 4 ATP. So that means the last step, that's where we're really going to get a lot of ATP. So where did all the energy go? To the NADH. So NADH and the FADH2 are what are holding on to the energy at this point. So we're counting on these in our last step in our stage 4 to get the energy out of and make finally our ATP. In order to understand the last stage, remember protein complexes. So you have a protein complex here. That just means that it's made up of 2 or more proteins. And so you can see here, this is 1 protein complex. And when you're looking at our last stage here of cellular respiration, there's actually 4 important protein complexes that are involved in our last stage here. So here's 1 protein complex, here's 2, here's 3, and here's 4. And we're going to see that when you look at one here, this protein complex only has 2 proteins that are part of it. This one has 3, this one has 2. So when you're talking about protein complexes, it's just an area that has 2 or more proteins that are a part of it.